Hello, science lovers. Welcome to the visual exploration of DNA replication. Today, we'll delve into 3 distinct modes of DNA replication, each unveiled through carefully conducted experiments. Let's embark on this scientific journey. In conservative replication, envision 2 DNA strands. As they come together, a new pair forms adjacent to the original, which represents the preservation of the genetic sequence. In the lab, heavy isotopes representing the original DNA sank, while new strands ascended. The layers forming in the CSCL gradient signifies the conservation of the original DNA structure. Transitioning to semi-conservative replication, DNA strands separate and pair with new partners. It's a partnership that ensures continuity, forming a harmonious connection between one old strand and one new strand. In the lab experiment, a distinct middle band emerged in the CSCL gradient. Now the DNA strands forms new pairs, and the partnership between one old strand and one new strand will be emphasized. This representation aligns with the concept of semi-conservative replication. In dispersive replication, visualize DNA strands dynamically breaking apart into segments, each segment joining with new strands. It's a meticulous process that creates diversity in the genetic material. Here you could observe that DNA strands dynamically breaks apart and mingle, creating a mosaic of genetic material. The CSCL gradient is appearing in a blurred pattern, signifying the dispersion of genetic material. As we conclude our exploration, the visual evidence from the CSCL gradient experiments firmly supports the semi-conservative replication model. The distinct bands observed align precisely with the predicted outcome of one old strand and one new strand pairing together. Our visual exploration concludes with a glimpse into the intricate world of DNA replication. If you found this journey insightful, we invite you to join us for more scientific explorations. Until our next visual journey, keep your curiosity alive.